All right, welcome back everybody. Now we are talking about biochemistry, chapter 3. So this chapter focuses on the non-enzymatic protein function and protein analysis. So we have some functions here of our proteins that are non-enzymatic. So that it would include structural function. So it helps compose the cytoskeleton. So these are some important ones to know. So collagen, elastin, keratin, actin, tubulin. So collagen will help with the strength and flexibility. Elastin helps with the connective tissue to become stretchy. Keratin is found in epithelial cells, and actin microfilaments are composed of actin. And tubulins, as mentioned, helps with the cytoskeleton. Next, you have some motor proteins, and they're responsible for muscle contraction and movement. So that would include dennin, kinesin, and myosin. And we'll go into muscle contraction in a later video. Then we have binding proteins, and those bind to specific substances to stabilize, to hold the concentration steady, or to sequester it into the body. Another group, and it's a little down here, hopefully you guys can see it. So another important thing, there we go, is our cell adhesion molecules. So those are proteins. Those include the cadherins, integrins, and selectins. And as well as our antibodies and immunoglobulin, those are proteins formed by beta cells. And so this is just a photo of the immunoglobulin antibody, just, it'd probably be useful to know. So we have the disulfide bond here, we have the light chains, the heavy chains, and here is the antigen binding site. All right, let's get back to our video. Let's just go over what the cell adhesion molecules do before we go on to the next part of this chapter. So as mentioned, the cadherins, those are calcium-dependent glycoproteins that hold similar cells together. Then we have integrins, and those have two membrane-spanning chains and permit the cell to adhere to different proteins in the extracellular matrix. And then we have selectins, and selectins allow the cell to adhere to carbohydrates on the surface of other cells. So that's most commonly found in the immune system. Those are just some important ones to know as well. So for protein isolation, a lot of this will be covered when we get to the organic chemistry videos. But for now, just a really, basic way of looking at it. So to isolate this protein, you can use electrophoresis, which uses a gel matrix, and you observe the migration. You can use native page, which maintains the shape. And then you can compare the mass to charge ratio. You can use SDS page, which denatures the protein. You can use isoelectric focusing, which separates the proteins by PI, and they allow the proteins to migrate. And then the chromatography has a lot of different types of chromatography, but it separates protein mixtures based on affinity. So you have column chromatography, ion exchange chromatography, size exclusion chromatography, and affinity chromatography. And all those will be we will delve into all of those in more detail a little bit later. But those are just some things to keep in mind of how you can isolate proteins. Now if we come up here, we're going to talk about biosignaling. So biosignaling, some two really important things to make note of are the ion channels and our enzyme-linked receptors. So the ion channels, you can have ungated channels, which means that they're always open. You can have voltage-gated channels, which means they open within a range of membrane potentials. And we'll see how that's useful in later biology videos. Then you have the ligand-gated ion channels, and that is only in the presence of a specific binding substrate will it open. And another thing is the enzyme-linked receptor. So a great example of that is the GPCR. So G protein coupled receptor. And that has a few different units here, and it is way more complex than what is pictured. I would definitely recommend looking up another video to explain this in detail if you would like to know. But just this is briefly covered in this chapter. So basically, you know, for example, when epinephrine binds to this GPCR, the beta and gamma subunits come off, and the GDP and alpha subunits can actually separate and bind to adenylyl cyclase, and then that will cause a cascade of reactions with cyclic AMP. So that is just an important concept. But yes, definitely look at another video for more detail on that. And yes, I believe that is the important parts of chapter 3. And we have covered a lot. So if you have any questions, leave them in the comments below. And other than that, stay tuned for our next video.